is November the 11th. Okay, so after today's lecture, this section is done. So Thursday and the next week, Tuesday, I both will do the review. If needed, and we will be complete a review and then move on to the final exam sections. Okay, so if possible, we could even have our last day of the lecture could be a choice for the final exam. If some of you want to take it early, plus like Friday and also the normal exam time. So just want to let you know about that. I already have practice exam 1, 2, 3. I already put on the eCampus, so you can take a look. Okay. So we want to continue today, talk about regulation and lateral operant, all those things. And still I'm going to write on the blackboard, we're going to take more notes. So we're going to finish talk about here, start writing. Okay. Okay, so first of all, we talk about, in this section, is central dogma. Remember, we had DNA goes to RNA, and RNA goes to protein. And then we talk about the DNA replication and DNA to RNA is transcription, and RNA to protein is translation. These we finished to talk about already. We left it one haven't talked. Remember, when the central dogma comes out, some people say it's wrong because it could happen the opposite direction from RNA to DNA. This is called the reverse transcription. Now, why they find it that? How they initiate this concept? So we want to talk about that, okay? So here is the thing. This concept comes out actually, which is how we say it. It's related to something we call we try to do a cloning or the DNA recombination. So let's say I have a circulating DNA sequence. This is from a bacteria. We also call it a vector. We'll talk about this later. Okay, right now, I have some sequence. This sequence right here. Let's say human hemoglobin. Human hemoglobin is like this. You have the alpha subunit, beta subunit. It's a tetron. Okay, I want to do a recombination. So I'm going to add it. Of course, you know the different ways, the restriction enzyme and the cuts and combined using gel electrophoresis to identify all kinds of them. Okay, let's say I combine. This is the part of the human hemoglobin DNA. Okay. I will combine. I want to do transcription. I want to do protein. Okay, I'm going to go this way. Same as central dogma. Then goes here. The protein. So the first question you're going to ask yourself, is this going to work? The answer is no. Why? The DNA is bacteria. Is a precariotes. And the human hemoglobin DNA, or animal, it is a eukaryote. they cannot naturally combine together and then go ahead to do transcription to RNA and the protein. You know why? Remember, for human DNA, when they go to the RNA, when they do the transcription, what they have to do? Do you remember? We talk about post-transcription modification. Remember these? And I draw the blackboard on the side last time when I talked, last Thursday. What are these? This is called the exon. And the blue one called the intron. Okay? This is for the eukaryotes, including human animal cells. Once this happens, what are you going to do? You have to cut them. All the intron, you have to slice or splice. Okay? You have to slice it. Then, all the exon has to combine. Remember, we talked about this. But my question is this process can do with the bacterial DNA? No, because there is no cell machinery to do the job, for bacteria to do the job. So this is not going to work. If the transcription is failed, the translation, of course, it's not going to work. So what are we going to do? If this failed, what we have to do? We have to do is based on, we're going to get a human hemoglobin, not the DNA. We're going to get the mRNA. Okay, let's say the mRNA is like this. We have like T. Let's say this is 5' to 3'. This is mRNA. So we're going to using mRNA as a template to synthesize a DNA. That's called a reverse transcription. Okay, we synthesize the DNA like this. Once you synthesize what you're going to do, this is the DNA. Once you synthesize, it's a hybrid. You have one band is mRNA, another band is DNA. Then what you're going to do? You have to license these. This rely on RNase. And then, well, we need some RNA as a promoter. Then what we're going to do, we're going to base on complementary sequence, we're going to seal these. How are we going to seal them? DNA, polymerase, plus ligase. So, based on these, what we get is actually a complementary DNA library. That's what we got. And then, we could to do transcription translation following that. This is what reverse transcriptase comes from. Now, of course, the reverse transcription is an enzyme called the reverse transcription. Transcripts what they come from, what this enzyme comes from. This reverse transcriptase comes from a virus, which is called a retrovirus. A virus isolated the enzyme from it. An enzyme isolated from the virus, which is called a retrovirus. Okay, so this is the one. When we finish talking about here, we will explain the central dogma. And remember, a long time ago, when it first comes out, they say it's nucleic acids become protein. They did not say RNA, DNA, RNA. And the reverse transcription, these are later people added. Okay? So this is something we added when we finished talking about central dogma. Okay, next couple of slides, we're going to talk about is the regulation. We're going to talk about the regulations. There is a lot of the regulation for gene expression, transcription translation. So we were using lactose operant as an example. Okay, first a couple of slides, how we understand it. So let's start. So we're going to talk about gene regulation. Okay, what is the gene regulation really talk about? So ideally, we think the DNA sequence is linear. And there is a places, we call it P. What is that? Which means per model. Now, per model, you need to know, there is always a conservative sequence. We mentioned about -35 and -10 pyramidal box, which means it's responsible for most of the capability and the function of the bacterial DNA. And remember, we talk about the flagella rotation, chemosmosis, that might be different. Okay, so let's say the promoter is. What the promoter is used for? We can do a brief review. This is rely on, going to be combined with a big chunk of them, which is we call RNA polymerase. And this RNA polymerase, how we recognize the promoter, rely on the sigma factor, let's say sigma 7. Okay, once they combine, what they can do? They can start to do transcription. Okay, always remember, this is from 5' to 3'. Now, what means the gene regulation? This happened, the combination, the RNA polymerase combined with the promoter is not that easy. They're going to be controlled by some factors. They could have something happen on the right side. Therefore, we have a position called O, which is an operator. They also could happen something on the left side. And then the position, we usually call it activated protein combined site. So I'll just write it called A. It's easy. You can understand this. Promoter and RNA polymerase, they combine together is not that easy. They controlled by left and the right side. This is the same thing you imagine, like you have a girlfriend or boyfriend, you guys get together, it's not easy. From dating to get married and finally have a baby and spend the rest of your life, it's not easy. So you know it's the same for bacteria. The two items combined together is always not easy. There are lots of factors to control. So that's easily understand. Just gave you a joke, so it's easy to understand. So then, look at these slides. It is complicated. What is called a positive control is a repressible gene. Or then it's come from like a negative control inducible gene. These are two, confusing you. It's like a twist of your mind when you first see these things. So how do you understand about these? On the right side, we always call, this is downstream. On the left side, it's facing you, okay? This is upstream. Okay, let's use a promoter as a sender. Anything happen on the right side of the promoter, which is attacking this O or leave this O, which is a downstream. This is we call its negative control. Anything happens on the left side, which is positive control. Okay, then both negative and positive control have two categories. It could be inducible inducible, or it could be repressible. Here the same thing. Inducible and or repressible. Now, of course, there's a gene for that, so we call it a gene. So it's very simple. This is the whole story. We talk about positive and negative control. If you draw this on your notebook, it's very easy. Using promoter and the center. Right side is downstream. Anything happens, we call negative control. Left hand, this is the upstream. If anything happens there, is called positive control. Now, every control has two categories, which is going to be inducible or repressible. What their function? They're going to bother the RNA polymerase and the promoter combine, or they let them combine. Either way. So this is the whole story, what's called the gene regulation. Okay. Now, of course, for the bacteria, the factors depending on their growth situation, their environmental situation, their nutrition requirements, and the growth status, all these things could be a factor. So that's why we're going to give you some of the examples. Okay. Now, first of all, we're going to talk about the lactose operant. This is the stuff we talk all the time. So I'll just draw a circle right here and make sure this is the overall picture. Now we're going to talk about the lactose operant. Okay? Lactose operant. Now, first of all, what is lactose operant? Or we just say lactose operant. Now, we want to make you a little bit easy. Okay, so I want to draw DNA sequence like this. What it is. But first of all, this is a 1967 Nobel Prize by two French scientists. It's a very magic stuff in the world. What we are talking about here is almost all of them are the Nobel Prize. Let's say DNA replication, that's called the most beautiful experiment in the world back in 1960s. This is another Nobel Prize. People find the lactose operant. There's a lot we can talk. I twisted your mind a little bit and related to all the knowledge we have learned so far. And I'll tell you. So, I want to make sure it's a little bit simple. I don't want to make it too complicated. The center position is called a P. That's a promoter. On the right side of position is O, is operator. And then here we have a position, I'll just say CAP. Cap position. Okay? Now, down here, you have some functional gene. What are the functional genes? Just the three blocks. Okay, what are they? It's called LAC-Z, LAC-Y, and the LAC-A. Now, some of your textbook, if you go genetics, biochemistry, they have something right here called ACI, LAC-I, LAC-N, whatever. Something they separate. They say the P is between O. We don't go those too difficult stuff. Okay, that's too complicated. Just want to make sure it's easy like this. Okay, what is CAP? Catabolite activator protein. Now, I usually say it's a binding site, protein binding site. What is P? It's promoter. What is O? Operator. Okay, then what are those three functional genes? What is lactate? Beta glucosidase. What is LAC-Y? I'll just take on the permease. What they say beta glycoside is permease. Then what is like A? You can say it's unknown because we don't know what the function it is. If you really say this is called transacetyl-lase, whatever they call it, we don't know the function. So more important is these two. Okay, this is the structure. Now, before we talk about the lacto-operon, you need to know some of the basic story behind it. Then you understand it. Okay, here is a bacterial cell. A very large bacterial cell. So this is a bacterial cell. I have a lactose coming. Let's say a lactose coming. Go inside. Well, they come in. You either pay the ticket or you let you in, or with some help. So they need a permease. You can understand like a permit that you come in. Okay. Then, what is lactose? It's very simple. First question, what is lactose? This is a disaccharide. What is disaccharide? Double sugar, if you don't know. So here's the sugar. What do we mean? What is double sugar? Glucose. Okay, then what's the structure of lactose? I'll give you extra points. You need to know what is glucose, is that right? Okay, what is glucose? How do you got glucose? You should know it's very clear right now. Okay. Goes down, goes up, goes down, up, and then up. That's glucose. Okay, so let me write a G there. That's glucose. Okay, what is the lactose? I said it's double glucose. Oh, you're going to write another glucose. Okay, very simple. So far, you should know what is glucose. If you don't know glucose, you're going to have trouble. So, write another glucose. OH. How they combine? Very simple. Something happens here. Don't want to make you too complicated. This is glucose. Oh, this is lactose. Combine it, become lactose. This is a very important concept. It's a glucose. Okay, then let's say bacteria can use glucose. So what are they going to become? This cell has to draw a little bit large. Okay. What are they going to become? If they have galactosides, they're going to become glucose, of course. So they separate, become a glucose. Lactose hydrolyzed by galactosides become another glucose. Okay? Then what else? They will generate isomer products, which is very, very similar to glucose. Only this position, the 4-carbon hydroxy group is down. What is this called? Galactose. Okay? Then, you also need to know, in this bacterial cell, this lactose could be generated a limited amount, a very limited amount, go to the side, will generate another product, which is called allolactose. So the same thing, you draw two glucose. What is allolactose? A little bit different from here. They are connected. What is this? Allolactose. This is a limited amount. Little amount. Or we say side products. Okay, you need to know, this is inside of the cell. What's going to happen? This is you need to know. When the permease laden come in, that's what's going to happen. Okay, then we're going to go ahead to talk about the story. First of all, when the research they do, to get a Nobel Prize, they're not going to do everything at the beginning. I covered everything. They only talk about is on the right side. So they only talk about is a negative control. Okay, so what is that? We're going to draw right here. Okay. We have